Alright guys, we're going to start off Unit 6 now, I guess this would be, with a discussion of molecular genetics. That's a fancy way of saying DNA structure and replication and transcription and translation. Basically everything that I just mentioned there encompassed into kind of this overarching thing they call molecular genetics. Okay, so it's a little bit more difficult in terms of we're going back to some of the chemical-based stuff that we talked about in first semester, which sometimes can give people trouble. The good news is that you know a lot of this stuff already. This stuff is information that you've hammered on quite often. Ever since you were little kids, you've talked about DNA structure. So a lot of the base knowledge is going to be easy to remember. Okay, so we're going to use that, this first video here, as kind of a way to do that. Just nothing more than to review some of that structure, get a little bit deeper into some of it, and use it to set up some of the other things that we're going to learn in this chapter about DNA replication. What I want to first do is kind of establish the fact that DNA, at one point, they didn't know a whole lot about DNA, obviously, right? We just kind of take it for granted now, but at some point, they did not even know that DNA was the genetic material. Okay, they did not know that it was the material that was passed from one generation to the next to pass those traits on. And so, some smart scientists had to figure that stuff out. And so, we're going to talk about one of them, at least here, to help us understand the fact that this experiment here by Frederick Griffith in the 20s was able to kind of shed light on the fact that it was DNA. That did play a role as the genetic molecule, the molecule that passes traits from one generation to the next. Okay, so what Griffith did, he worked with strains of bacteria, and it turns out that bacteria and viruses and things of that nature are really good organisms for genetic study, specifically these molecular genetic studies, because they're very, very simple, they're easy to grow, and they breed very, very quickly. Okay, so he worked with these two strains of bacteria. One was a pathogenic S strain. He called it the S strain. And one was called the harmless R strain. Okay, so one of these bacteria would cause disease. One of these bacteria would cause no disease. Okay, and he figured out that DNA would cause transformations in one cell that maybe didn't have a specific set of traits before its transformation. Okay, so this term transformation is one that we're again going to come back to eventually, I think later on in this unit, when we talk about bacterial transformation, and this is our first introduction into it. So the term transformation means a change in genotype and oftentimes phenotype due to assimilation of foreign DNA, and bacteria do this a lot. And so this was how he was able to understand that there was something in these cells, these bacteria cells, that was being transferred from one cell to the next, and then as a result caused disease. So let's just briefly talk about this experiment that he ran, because it is relatively simple. He took, we're going to talk a little bit about the controls here as well, and some experimental design stuff, but he took these living pathogenic S bacteria. Okay, they're indicated here by the orange kind of outline. Okay, so these bacteria are pathogenic. If they go into something, they make it sick, and then it dies. And so that's what he did here with the mouse. He put this into the mouse. He knew right off the bat that these pathogenic S strains should kill the mouse. Okay, and lo and behold, it does. That's why it's a control. We kind of know the outcome of that particular test already. He then took the other strain of bacteria called the living R cells, okay, the ones that if you get them, they're not going to make you sick and they're not going to make you die, and he put them into a mouse, and lo and behold, that's exactly what he expected. The mouse was going to be healthy, okay, and it was, because these things don't cause disease. The third part of the control here is to take the S cells, the pathogenic S cells, and just kind of kill them. Okay, with heat, you can kill these things, and when you kill a pathogenic strain of something, it's no longer pathogenic. It's not gonna kill you. It's not gonna make you sick anymore. So he took these heat-killed S cells and he put them into the mouse, and that's exactly what happened. The mouse was healthy. He figured that was what it was going to be, and that's what it was. Now for the actual experimental part of the test. The experimental group, he took a mixture of heat-killed S cells, okay, and those cannot cause pathogen. They cannot cause sickness, okay, because they've been heat-killed. And he took living R cells, okay, living R cells. These are cells that he saw do not cause disease in the mouse, and the mouse lives, right? So in theory, this is what he's thinking. Okay, I've killed these S cells. Okay, they're not going to cause disease. These other cells are not harmless. They're not harmful, right? They're harmless. So when I inject both of these things in a mixture into the mouse, what should happen? What should happen is that the mouse should live, right? We've got harmless cells mixed in with dead cells. The mouse should live. So we did it, and lo and behold, the mouse died. And this is where we start to see how these kind of discrepant events help us understand and learn things, right? He saw that the mouse died, and so it totally went against everything that he thought was going to happen. And what he did is he went into the mouse's body and, extracted some blood samples and things like that. And what he found was that there was actually living S cells in the blood sample of the dead mouse. But where did they come from? They weren't in the original samples. He only put harmless ones and heat-killed ones. Okay? What had actually occurred is that these heat-killed S cells still had pathogenic DNA inside of them. And what actually occurred is that that pathogenic DNA was transferred into the harmless cells and transformed them, changed them into something that was now pathogenic and able to kill a mouse. So what we actually have happened, these cells right here, when they're heat killed, because they're dead cells, that's why they don't cause disease. They still have the DNA that causes disease, and that DNA was then transferred into these harmless cells. Okay, so kind of a neat little experiment that kind of showed that there was some kind of substance that was being transferred from one cell to the other, and it conferred those traits from the one cell and gave them to the other cell. Okay. Now, it wasn't until 1944 that a group of scientists, Avery, McCarty, and McLeod, announced that the substance that was doing the transforming was DNA. So it even took another, 16 years after Frederick Griffith's experiment until they were able to unequivocally point to the fact that it was this molecule that they called DNA that was causing the disease, that was the hereditary material. Okay. Now there's tons of other examples of experiments that help to illustrate that DNA was the genetic material and that it was the thing being passed from one generation to the next. This is just one that kind of helped kick it all off, right? There was something being transferred between one cell to the next, and that thing ultimately turned out to be DNA. Okay. Now, we talked a little bit about here bacterial DNA, which is known as prokaryotic DNA. Okay? One thing we have to establish before we continue to move on through this material is that bacterial DNA and eukaryotic DNA, eukaryotic DNA is like ours, they are fundamentally different. Bacterial DNA is usually only one chromosome, and it's usually circular. And so I've shown a couple, there's the artistic rendering of the two different types of chromosomes down here, and then an actual microscope view of them up here. And so up here we see the prokaryotic DNA. It's a circle. Obviously, if you spread that apart, it would be a circle, right? And then the eukaryotic DNA over here is basically just linear. It's a line. If you pulled it out, it's like a string. It's a line, and it has a beginning point and an end point, and there's usually a lot of them. Okay. So they are contrary to what we see in the bacteria. Eukaryotic chromosomes, much more complex, and many more of them. Okay, so do keep that in mind as we work our way through this material, and we're going to revisit some of the stuff about bacteria a little bit later, hopefully in the unit. Okay, so let's get into building a structural model of DNA and talk about some of the people and things behind that, as well as just kind of get into the nuts and bolts of it, right? Some of the names that you're going to see here are familiar or should be familiar to you, and some of them we're going to delve into a little bit more because they kind of got the shaft back in the day. So we'll spend a little bit more time talking about somebody named Rosalind Franklin and her discovery. So Maurice Wilkins and Rosalind Franklin worked in a field of molecular experimentation called X-ray crystallography, which is it's actually quite a fascinating field. I know a little bit about it, and there's a lot to know about it. But it is a way of imaging organic or biological molecules. And so Rosalind Franklin and Maurice Wilkins set out to ultimately image and thus deduce the structure of DNA based on taking an X-ray picture of it. Now, that information was ultimately claimed by Watson and Crick, and you guys know that, right? But there are stories that Rosalind Franklin had kind of settled on a very similar type of model to Watson and Crick, and they stole her data and this, that, and the other thing. There's a lot of stories about it. So some of what we're going to do next week is to read up about it and do a little bit more research about it, and we'll talk more about that in class. But anyway, these pictures that Rosalind Franklin took of the DNA allowed Watson and Crick and herself to deduce the fact that the DNA molecule was in fact helical, it was double-stranded, and it was connected by what were called nitrogenous base pairs. Okay, so there's a picture of Rosalind Franklin, and this is a picture of her DNA model. Okay, and one of the things that I would like to do is get into how this is DNA. How this is DNA. How in the heck did this picture, when she looked at it, this X-ray picture, how did she deduce that that was DNA? What we know to be DNA is, I'll get to it here, that picture. Okay, how did looking at that thing that looks like an S, that big blobby black and white X, allow her to understand that this is what she saw? Okay, but she's an expert in her field, and they know exactly what those pictures mean, and she was able to kind of come to the same conclusion as Watson and Crick. Ultimately, they're the ones that got the credit for designing this relatively elegant double helical model for that DNA structure, and the fact that it was connected by base pairs. And so that's those two right there. All right. So from here out is going to be kind of some, I would say, some review more than anything, because we don't need to get into the intricacies of how they discovered, the base pairs, or the fact that there was a sugar phosphate backbone or anything like that. We're going to talk a little bit about how they were able to discover what was paired up and how those pairings occurred. But let's just talk about some basics of, hopefully this works. Yes, I got the right one. Let's just talk about some basics, or review some basics about the structure of a DNA strand. So what we see on the picture right here is a single strand of DNA, and obviously we know DNA is double-stranded, but this is put on here just for simplicity. And one of the things we learned back in Chapter 5 was that DNA is effectively a polymer made up of monomers, right? And each one of those monomers, which I'm outlining right here now in red, as I'm doing this, why don't you guys go ahead and try to guess what those are. Pause the video and try to guess. Or just know, because you should know it. And I just realized that it's on there. Okay, they are nucleotides. Okay, so the larger polymer of DNA is broken up into monomers of nucleotides, which is what I just outlined in red. Okay, so each one of those things that I outlined in red has a specific set of molecules or parts to it, right? We see that each one of them has the phosphate group, the sugar. This is why they call this the sugar phosphate backbone. All this stuff that's kind of outlined in blue would be the quote-unquote backbone of the DNA. and then is made up of one of 4 specific nitrogenous bases, okay, which we classify as either what are called purines or pyrimidines, and we'll talk about the difference between those in a second, okay? There we go, get rid of that stuff. Okay, one of the other things that we need to establish here, and I think again that I kind of did talk about this at some point, we need to establish that DNA is directional, okay, that it has direction, that it has ends to it. When we see these labeled as what are called the 5' end, I'm going to use green, the 5' end and the 3' end of the DNA strand, okay? Now the 3' end and the 5' end are labeled that way based on the sugar molecule that makes up part of the sugar backbone. And so what we see up here at the top is that sugar molecule, and if you kind of, can I zoom in here? This is something new. All right, perfect. All right, so what we see, go through that again, is that each corner of this little sugar molecule, plus this right here, is labeled with a number, okay? And each one of those corners represents a carbon molecule, or a carbon atom. And so this carbon atom is labeled as the 1', this is the 2', the 3', the 4', and then the 5' up here, okay? That's just how they do it. That's one of those skills that when you take organic chemistry, they'll teach you how to number the carbons in a big organic molecule like this, and that's how you do it. And so the name of this end of the DNA molecule is named for the fact that this 5' carbon, okay, the one that I outlined right here, this guy, is at the top. Okay, it's the one that's exposed. It's not really covered by anything, it's just out there open. Okay, now if we go down to the other side, the 3' end, again we can see that the corners are numbered, and they start with the rightmost one, and they label it 1, 2, 3', 4', and then the 5'. Okay, and what we'll notice is that it is the 3' carbon that is kind of out here all by itself, all alone, uncovered, unattached to anything other than this hydroxide group. And so that's why they call this end the 3' end. Okay? And that's going to become important when we start to talk about DNA replication, because new DNA base pairs are only able to be added in a certain fashion. Okay? And it's based on which carbons are open. Is it the 5' or is it the 3'? Okay? All right, so I mentioned that the 2 strands of DNA are connected by nitrogenous bases. This is one of the things that Watson and Crick actually kind of attribute to this discovery. And we notice that those things are right in the middle. Okay, the nitrogenous bases are right in the middle. And we would have another strand of DNA over here with their nitrogenous bases kind of coming in to meet. And we notice that all the pyrimidines have a single ring, and all the purines have a double ring. Okay? Now, one of the things that was noticed in the analysis of these X-ray pictures is that the DNA molecule was of uniform width. Okay, it had uniform width, meaning that everywhere you looked, it was the same width no matter what. It didn't do one of these like an hourglass or something like that. And so what they had to do was figure out how did this molecule, they knew that there was these nitrogenous bases, the purines and the pyrimidines, and they knew that they were in the middle, but they had to figure out how they matched up. Okay, they had to figure out that a certain way of putting these purines with the pyrimidines, the A's to the T's or the G's to the C's, would yield a certain width, quote unquote, width of DNA molecule. Okay, so if you paired up a purine with a purine, okay, so 2 double ring structures, it was too wide. It was wider than what they had measured in their experimentation. If you paired a pyrimidine with a pyrimidine, the single ring structures, it was too narrow. It was narrower than the actual experimental width of the DNA strand. And if you paired a purine with a pyrimidine, a single with a double, then they realized, oh, it's just right. It's like Goldilocks and the Three Bears, right? It's just right. It fits the width that was measured perfectly, okay? So that allowed them to understand a little bit more in depth that it was the purines that matched with a pyrimidines. And it turns out that there was even more specific base pairing structure in that that A's always went with T's and G's always went with C's. And this was helped to be determined by a guy named Chargaff. Okay? And Chargaff did some analysis of the components of the DNA, and they knew the structures and how to indicate adenine or thymine or guanine or cytosine. They could test for those things. And what Chargaff did is he took a sample of DNA and analyzed it for the amount of adenine or A's in the DNA. And so he said, okay, I found that there's 7 billion bases of 7 million molecules or billion molecules of adenine. And then he looked at the T's. And he said, oh, there's 7 billion molecules of thiamine. Oh, that's weird. And he looked at the C's and said, okay, there's 3 billion cytosines. And then he looked at the G's and said, oh, there's 3 billion guanines. Huh. Well, I know that the A is a purine or a pyrimidine. I know the T is a purine pyrimidine. Let's match those up, right? See, I've even forgotten which ones are which. Okay, so he said that the pyrimidine T should match with that purine A because there are equal amounts of them. Okay, so there's one adenine for every thiamine. That's what Chargaff said. And that all these pieces of evidence allowed them to understand more intricately what was going on with their DNA model. Okay? So ultimately that's what you get. You get the thiamine, which is the pyrimidine, and the adenine, which is the purine, and the cytosine, the purine, and the guanine, the pyrimidine. Or did I see if I've done messed it up again? The adenine is the purine, and the thiamine is the pyrimidine, and the guanine is the purine, and the cytosine is the pyrimidine. You might want to go ahead and just try to memorize those, because obviously I haven't. But anyway, they match up, and there's a specific way that these two sets of molecules will come together, right? Adenine and thymine are always going to come together, and what you'll notice about them is they share 2 hydrogen bonds that keep them attached, whereas guanine and cytosine come together and they actually share 3 hydrogen bonds that keep them together. Okay? All of this evidence combined, knowing that there are 2 strands of this stuff, knowing the fact that the purine has to match with the pyrimidine, knowing the fact that there's equal amounts of A's and T's, all of these things put together allowed scientists to understand this basic double helical ladder structure of a DNA molecule, where you have these simple base pair rules. And all of this stuff really, as it gets down to it, is elegant because what it allows for is a way of replication. Okay, it allows for easy access to replicating the DNA, which is exactly what they were setting out to find. They noticed that cells replicate, right? And they noticed that each cell was genetically identical to the other one, but they want to know how. And they want to know what was the structure that allowed that. And it's this very simple, elegant structure that can actually be taken apart rather easily. We know that hydrogen bonds, like we see in the middle that hold these 2 strands together, are relatively weak bonds, right? So everything from the backbone to the little letters in the middle to the hydrogen bonds that put them together allow for a very simple mechanism of replication. So they got it right. There's a little bit of controversy entwined in all of this, and a lot of very smart people at some points working together, but they did get it done. And once they figured out the structure of this molecule, genetics was kind of unlocked at that point. There was new discoveries being made all the time. Okay, So as we transition into the next section, I'm going to put together a couple different videos on DNA replication, and those videos will be shared with you soon. So I will talk to you guys later.